Life Science. Hope you're doing well. Today we're going to continue Chapter 6, Role and Components of the Immune System, with Document 5, Lymphoid Organs. So, the objectives of this document, first of all, is to list the different lymphoid organs. Second is to identify the role of each lymphoid organ. And third is to explain the maturation of lymphocyte B and lymphocyte T. Let's first start with the organs of the immune system. As we have learned before in the previous documents, that the immune system consists of several organs and cells. And we have already talked about the cells that are the white blood cells. Let's remember, what are the white blood cells? What are the different types of white blood cells? They are granulocytes, monocytes, lymphocytes, and the mast cells. Okay, now, what are the organs of the immune system? These organs are divided into two groups. The first group, we call them primary lymphoid organs, and the second group, secondary lymphoid organs. What are the primary lymphoid organs by definition? The primary lymphoid organs are organs of the immune system where white blood cells are produced, and it's where they become mature. So, these organs are the site of production and the site of maturation of white blood cells. Let's see what are these primary lymphoid organs where the white blood cells are produced and where they mature. So, site of production and maturation of lymphocytes. In order to determine the site where white blood cells are produced and where they mature, and mainly the lymphocytes, experiments are carried out on 3 lots of mice, and these experiments are represented in document A. Look at these experiments. We have 3 lots, A, B, and C, of mice. Experiments are realized on every lot in a different way. The conditions of these experiments consist of doing irradiation and then grafting. What is irradiation? What is the aim of irradiation? Irradiation, it consists of treating specific organ with radiations, of course. And why? Because irradiation permits to destroy dividing cells. And since the bone marrow, as we saw previously in document 4, that it is rich in stem cells that are highly dividing cells. So by doing irradiation, we ensure that the cells in the bone marrow are destroyed. So when we use the irradiation as a condition in the experiment, it's just like we're telling you that the bone marrow is destroyed or there is no bone marrow. So let's see the conditions, the different conditions in lot A, B, and C. In lot A, irradiation followed by graft of the bone marrow. So the bone marrow is destroyed, then there is graft of the bone marrow. So at the end, the bone marrow is still present in lot A, that we can consider it as a control. Lot B, there is ablation of the thymus, and we're going to see what is the thymus. It's an organ located at the level of the chest, and it should be, of course, a lymphoid organ. So, Ablation of the thymus, followed by irradiation, followed by graft of the bone marrow. So as a result, we can say that lot B, they do not have a thymus, but they still have bone marrow cells. And lot C, ablation of the thymus and irradiation, then graft of the thymus. So irradiation means no bone marrow. Ablation of the thymus followed by graft of the thymus means that there is a thymus. Okay, now we have the conditions of the 3 experiments, and we have the results obtained for every experiment. The question is, interpret the experiments of document A. Dear life science students, I need you please to pause the video for a while, try to interpret these experiments on a piece of paper, and then let's correct it together. So, interpretation of these experiments. I'm going to remind you a little bit about interpretation. It consists of doing first an analysis, followed by a significance. In the analysis, what you should do is to relate the variable factor that you can find here in the conditions with the result. After relating the variable factor with the result, then we can reach the corresponding significance. So, in lot A, in the presence of bone marrow and thymus, there is bone marrow, and since here we're not talking about any experiment of ablation of the thymus, so the thymus is present. So lot A having bone marrow and thymus, there is production of LB and LT. While they are not produced in lot C when the bone marrow is absent. So the variable factor here is the presence or the absence of bone marrow. It is present in experiment of lot A and it is absent in lot C. Okay, so when the bone marrow was present, B and T lymphocytes were produced. When the bone marrow was absent, there is no production of B or T lymphocytes. So this shows that the bone marrow is responsible for the production of LB and LT. And this is the first lymphoid organ, which is the bone marrow, responsible for the production of LB and LT. Now, in lot B, in the absence of the thymus, there is production of immature LT and mature LB. So, in the absence of the thymus ablation, there is production of immature LT and B lymphocytes. It means that B lymphocytes are mature here. However, LB and LT mature when the thymus is present, as in lot A. Whenever the term mature is not mentioned, this means that B and T are mature. So B and T are produced and they are mature in lot A in the presence of bone marrow and the thymus, while in lot B with no thymus, only LB mature, while LT, lymphocyte T, are immature. This means that LT mature in the thymus and LB mature in the bone marrow. So, as a conclusion, the bone marrow is the site of production of LB and LT and the site of maturation of LB, while the thymus is the site of maturation of LT. The primary lymphoid organs are then the bone marrow and the thymus. They are the site of production and the site of maturation of LB and LT. You see in this figure, This is the bone marrow, this tissue found inside the bone, and where LB and LT are produced. Here is the thymus, small organ located at the level of the chest. At the level of LT, the immature LT that don't complete their maturation in the bone marrow, they leave to the thymus where they undergo their maturation. Let's now move to the paragraph about lymphocytes maturation. What is the meaning of the term maturation? A lymphocyte is mature, or we can say it is immunocompetent when it becomes functional. So, what do we mean with functional lymphocyte? A functional lymphocyte is a lymphocyte that is able to bind in a specific manner to a non-self antigen. Of course, a non-self antigen, which is an antigen that doesn't belong to your body. So every lymphocyte in your body that is able to bind to a non-self antigen is called functional and then is considered to be a mature lymphocyte. How? Let's see. In document B, you have, this is an immature lymphocyte present in the bone marrow and is waiting to undergo maturation. First of all, this immature lymphocyte, it gives rise to several lymphocytes. And as you see, every lymphocyte of these presents on its surface a different receptor. Every color indicates a specific type of receptor. So, if we're talking about LB, then the receptors are BCR, or antibodies, if you remember. If we're talking about LT, then these receptors are TCR. Okay, so do all these lymphocytes become mature? Actually, no. And this depends on whether the receptor can bind or cannot bind to non-self or to self antigens. What does this mean? Here, you see all these lymphocytes, each with a specific type of receptor. A remark to be said here that a specific lymphocyte carries only one type of receptor. Okay, these are examples of self-antigens, and these are also examples of self-antigens. If a lymphocyte can bind to a self-antigen, then this lymphocyte is said to be autoreactive. So what is an autoreactive cell? It is a cell that binds to self-antigens. The self-antigen could be, for example, a hormone present in your blood, or a protein, a self-protein found on the surface of a cell in your body. So, at any time, a lymphocyte with its receptor can bind to an antigen that you possess and that we call self-antigen. This lymphocyte should be eliminated. It is said to be autoreactive and should be eliminated. How? By clonal deletion. What do we mean with clonal deletion? It's the deletion of clones of lymphocyte of this type, for example. So this lymphocyte carrying this receptor in red, of course we have lots of lymphocytes like this one. So we say these are clones of lymphocyte. Clones, identical copies. So there will be clonal deletion of all the lymphocytes carrying this kind of receptor that can bind to this kind of self-antigen. Then what remains is only a lymphocyte with a receptor that cannot bind to self-antigen. Then these receptors can bind only to non-self antigens like those present on the surface of bacteria, for example. So we need in our body lymphocytes with receptors that can bind to non-self antigens. Of course, because if our lymphocytes bind to our self antigens, then they are going to eliminate our cells. And this is what we call autoimmunity. And it's a disease that we're going to talk. It's a malfunctioning of the immune system, of course, that can lead to disease. We're going to talk about this in chapter 8. So then what are the mature lymphocytes we have in our body? The mature lymphocytes are lymphocytes that are able to bind only to non-self antigens. So, for example, let's talk about the example of the maturation of B cells. Suppose this is a self-antigen triangle and this oval is a non-self antigen. We have in the bone marrow production of LB1 carrying a receptor that can bind to non-self antigen and LB2 carrying a receptor or antibody that can bind to the self antigen. Which one will be preserved and which one will be eliminated? LB1 is going to be preserved and to become mature since LB1 can bind to non-self antigen and we need this lymphocyte because we might one day catch an infection with this non-self antigen. However, LB2 carrying a receptor that can bind to self-antigen, this LB2 is autoreactive and it should be eliminated. So, all lymphocytes whose receptor can bind to self-antigens, then they are autoreactive, they are eliminated by clonal deletion, while all lymphocytes whose receptors can't bind to self-antigens, these are preserved. So, to sum up, the primary lymphoid organs are divided into bone marrow and thymus. The bone marrow, it's where LB and LT are produced and where LB mature. The thymus is where LT mature. Thank you, dear students, for your attention. Take care of yourself and see you later.